Well, hello to you all, and thank you for your interest in Labroots and in this particular conference. It is a privilege for me to participate, and I also wanted to thank Labroots for organizing these events and for scheduling these educational webinars. So as was said, my job today is to tell you about our experiences with testing tissue chips. As you can see on the screen, I'm a toxicologist by training, and I also have an MD. And I came to interacting with tissue chips as a biomedical researcher and a mechanistic toxicologist. And before I begin, I wanted to have a disclaimer. A couple of years ago, I was interviewed by Nature Medicine for a feature article that they were putting together on chip power. And a couple of quotes that they used in that particular article reflected how I felt about tissue chips and microphysiological systems 2 years ago. And in part, my presentation today will be a transition of me personally as a scientist and as a toxicologist, and what type of confidence I was able to build into myself and into the stakeholders that we collaborate with. So I said that I don't want to assume that more complicated system is better. And as you can imagine, tissue chips are much more complicated than traditional in vitro systems. And I also said that I was at that time a healthy skeptic about tissue chips and how much promise they can actually hold. And I did feel that a number of developers have overpromised with respect to what they can deliver. And this was something that we've probably all seen before, with promising technology not living up to its expectations and perhaps fading much sooner than it can really deliver because the expectations were much greater. So what we have tried to do in the last couple of years is to help both the bioengineers and the end users to have greater confidence in tissue chips as a technology that can be used for safety and toxicity testing, and also in other parts of drug development. So before I begin giving you the data, I wanted to thank National Center for Advancing Translational Sciences for funding us with a 2-year grant in 2016. And this year, or late last year, they actually funded us again for 2 more years. But all of the data that we have been able to collect as a tissue chip testing center at Texas A&M University was really a product of 4 very able postdocs and technicians. And then we have a couple of people that are helping out in analytical chemistry, and I'll share some of that data with you as well. And most importantly, we have a number of faculty who we are able to reach out for consultation. There's really myself and Dr. Clifford Stephan at one of our partner organizations that have overseen the laboratory experiments. But most important, I'm very grateful to our collaborators from 9 universities that have shared their devices with us, that have hosted our staff in their laboratories to learn how to use their technology, and who have stayed interacting with us with respect to protocols and data analysis and interpretation of their results. So I'm really grateful not only to principal investigators, but also to their students, technicians, and postdocs who have really gone out of their way working with us. So I wanted to start at the very beginning, and this slide is the courtesy of Dr. Danila Tagli from NCATS. And NCATS is one of the funding agencies that is part of National Institutes of Health in the United States that has been funding a lot of biomedical engineering research into developing tissue chips. So what you can see at the bottom of your screen, NCATS has provided a number of grants, and I will tell you more about them on the next couple of slides, that are aimed to develop microphysiological systems that replicate organ function and structure that can be used to test compounds that can be actually taken outside of the laboratory. And these particular devices are meant to be beyond just the publication and something of an academic interest. They're really meant to be used by the stakeholders, and that's what you see on the very top. The industry and regulatory agencies really are the ones that are prospective users of this technology. And most importantly, this particular technology, if people have confidence in it and if it really works, can be something that contract research organizations can provide as a service to multiple clients to build up the data and to increase further confidence in using these types of data for decision-making in regulatory environments. However, as frequently happens, there is a large gap between the people who develop technology and people who can be potential users of this particular technology. And that gap in biomedical sciences is very challenging to bridge. And NCATS designed a program to bring tissue chip testing centers who would be trying to be the bridge between the technology developers and the technology users, because there's a lot of writing, not only in terms of finances, but also advances of drug development and chemical safety evaluation on this. So NCATS issued a program announcement in 2016, and they called for proposals that would last for about 2 years, that would test 2 to 3 new devices about every 6 months, and would have a throughput of a couple of compounds and tested for a couple of different phenotypes and replicated a couple of times. So really, 2.5 years ago, nobody knew what is the opportunity and whether this tissue chip testing is even possible. So NCATS has called this program announcement Next-Gen Tissue Chip Testing Centers, Validating Microphysiological System. And validation means different thing in different contexts. What you can see on the left at FDA, analytical and clinical validation is really something that is quite established. I'm using, excuse me, an example of U.S. Food and Drug Administration, but there are very similar principles in Europe and in other countries. And validation really is broken into both analytical and clinical validation. And in both of these contexts, one really needs to understand how the device or how the test will be used. And once you understand the context of use for that particular device, you can understand where the bar is for how good that technology should be. And once you understand the use and what the level of evidence one needs, you can actually embark on the process of qualification. And qualification of biomarkers, qualification of survey instruments and other things in drug world is not an easy process that takes years, and it takes many stakeholders. Now, on the chemical side, there's validation of alternative methods that many of you may be familiar with, and that's a process that is very rigorously regulated and specified by a number of legal documents and frameworks. In Europe, for example, the Cosmetics Directive and the REACH regulation describe what validation alternative methods really is. And then United States, Japan, Korea, Brazil, and other countries, there are very parallel efforts. And internationally, Organization of Economic Cooperation and Development, OECD, has actually a guideline that is a guidance document on validation and international acceptance of new or updated test methods for hazard assessment. And that guideline states that there are 3 components of 3 major elements in validation. There's a testing of reliability, there's a testing of relevance, and then there has to be a purpose in which this particular test will be used. And so when this comes to tissue chips and the vision that NCATS had 2.5 years ago for what tissue chip testing centers should do, it was not entirely clear which ones of these qualifications or validations, or is it reliability or is it relevance? And it was left to the grantees or people who wanted to solicit for these particular grants to define what that meant. So first thing that we had to start with was to define what tissue chips are out there. And NCATS specified that the tissue chip testing centers were only to work with developers who have been funded by NCATS. And in 2012, there was a first round of grants awarded, and all of this information is available on NCATS website. If you just Google NCATS and tissue chip, you'll be led to a website that has plenty of information on the developers, on what they've done, what they've published, and a lot of information is provided for you. But as you can see, there is a diversity of institutions, there's a diversity of the projects, of tissues or models that have been proposed. And we reached out to all of these, and then we ended up with about 2/3 of the developers who were interested in working with us. And as I will say at the end of this presentation, one of the challenges is to actually convince people who develop technology to let a third party test that technology before it actually hits real world. And that's just the reality, and I'm not really taking sides in this myself. Now, when we proposed to do tissue chip testing center, and as you can see, we have a TaxVal as our title, and we're at the Texas A&M University in College Station, Texas, we really reasoned that this is it takes a village to do this type of work, and you need to have experts from different disciplines. And as you can see, this puzzle board at the bottom left, you need to have expertise in toxicology, analytical chemistry, and bioengineering and microfluidics. You need to have folks who understand how to use these data in pharmacokinetic modeling. There are a lot of measurements that are being taken that are very diverse, from protein measurements to gene expression to images. And then you have to do this at GLP-like environment, so you have to make sure that your work is actually quality controlled as well. But there are other parameters and core principles that were important to us. First, we really wanted to make sure that our team of scientists is independent of the NCATS funded tissue chip developers. So as you can see on the next click, we have a diversity of faculty, and even though some of them are bioengineers, they're not funded currently by MCAT, so we really had no conflict of interest as far as we are concerned. Second important thing that I wanted to highlight is that you really need to compare the data from the 3D microphysiological system to the standard in vitro culture model. It is also important to compare to the in vivo situation whenever possible, but because in principle, these microphysiological systems are using cells, then recreating some form of a much simpler system that probably has a higher throughput and can be attainable by more laboratories was something that was very important to us. And unfortunately, in the bioengineering world, the tissue chip development world, very few labs actually provide comparison to the 2D systems. So we had a complicated org chart, and I'm not going to take you through it for a very long time, just to point out that not only it takes a village of a lot of different people with a lot of different expertise to have this type of a throughput to test 8 to 12 platforms in 2 years, you really need to have capacity to do this. And so we were fortunate to have 2 different sites at Texas A&M University, one in Houston, one in College Station. It was about an hour and a half drive between these 2 facilities. We had 2 labs in Houston and 4 labs in College Station that kind of came together and interacted on a weekly basis and exchanged compounds, exchanged data, exchanged protocol and information. But we also were interacting very closely with our tissue chip developers, as I already mentioned, but also that's not the end of it. We had monthly teleconferences with our funder and CATS, but also with representatives of various pharmaceutical companies that have since organized into the IQ consortium affiliate on microphysiological systems. And last but not least, all of the data that we're generating, we're depositing into the University of Pittsburgh Microphysiological Systems database. This was one of the grants awarded under this mechanism. And I, again, welcome you to go and explore Microphysiological Systems database. Again, you can just Google for that and you can easily find it. Now, as I already mentioned, this type of an effort that I will describe to you in some detail took only about 2 years. And for those of you in academia, you certainly would appreciate that 2 years is a very short period of time, because by the time you actually get funding in and actually start projects, hire staff, and actually begin doing things, you already lost valuable time. So we had to devise a standardized workflow that we can follow, not only for every chip, but also to organize our work. And I already mentioned that we try to test devices in about a 6 months period, because we reasoned that in the real world, whether it's a pharmaceutical or a chemical company or contract research organization, people who are not developing the particular model will not have long enough staying power. If it really doesn't work in a couple of months, then it's not worth investing more time and you need to move on to something. So we try to use very similar thinking process. But you also see that there are different tiers, there are different steps. The first step is really to establish a collaboration, and there's a tremendous amount of legal agreements that have to be negotiated between the universities to change the technology and data and do other things. You have to share protocols, you have to actually go in and send staff to train in the developer labs. And once you're through that, then you actually get the devices and you try to put it all together in your own lab, and then with without cells, try to see if you can establish flow or if you can do other things. And at that step, we usually started putting chemicals on these devices and examining whether or not we have a lot of binding of chemicals to these devices, because they're made of various types of materials, and many of these materials can bind compounds, especially based on their physical chemical properties. Then once we are through with that, we can actually try to replicate what the developers have published or were just about to publish, and we would develop in that process very detailed protocols and standard operating procedures. And then if we're through with that, through our conversations with our colleagues in pharmaceutical companies and other toxicologists, we would try to define some form of a context of use for each of these models. And we'll try to conduct additional studies that can gain additional attention from the potential end users, that can actually answer some of the questions, very practical questions that they have that may or may not have been addressed in the original publication that just described the technology itself. So I was very pleased that over these 2 years, we were able to complete 11 different tissue chip platforms. You can see on the left are the diversity of tissues and models. There's only one that was, similar, and I will tell you more about this. We had 2 models of the liver. All of these, as I already mentioned, were funded by MCATs. There are a lot of other technology platforms out there. There are other universities, there are other companies that developed that. We had to focus on what was funded by MCATs. We had collaborated with 9 different universities, and we have to complete a variety of legal agreements with them. And we've tested 7 of these in College Station and 4 of these in Houston. And as you can see, by the end of 2018, we've been able to take 10 of the 11 all the way through Tier 2, and then 1 of the platforms we stopped in Tier 1 because of the throughput and other challenges that we had. If you look at the timeline, more of a Gantt chart-like view, you see that we started in 2 sites. The maroon is College Station, the blue is our site in Houston, and we got confidence in working with the developers, getting into this workflow. And then our next period was more confidence with a couple of platforms on each site. And then towards the end, we really were confident in our ability to run multiple platforms in parallel. So we were able to work with 3 to 5 different platforms at the same time. So let me give you a couple of examples of what the data looks like. And I broken these slides into types of perfusion or lack thereof in these particular models. Because for me as a toxicologist, it was very interesting to learn that in the field of biomedical engineering, tissue chip or microphysiological is not really, very well-defined terms. And really each laboratory defines what is microphysiological, what is or is not a tissue chip themselves. And as long as they can convince peer reviewers and publish papers, and if they can convince funders to get more grants, that's how they perceive what is microphysiological. But obviously, each platform is trying to replicate either a tissue, either organ, or some part thereof, and then it actually is trying to replicate some sort of physiology or anatomy on the microscale. So a good couple of examples were platforms that actually have no flow. They're static cultures, but they are very complicated cultures. One is bone with and without tumor model from Columbia University. And what this is essentially a plate which has chambers, and these chambers contain in them pieces of decellularized bovine trabecular bone that have been seeded with human osteoblasts and Ewing sarcoma cells. So it's a human tissue, but there is no flow in that tissue. And what we have done after repeating the experiments that the colleagues at Columbia University did, we actually tried to do or simulate treatment of metastasis into the bone, and we tried to look at which drugs are used in the clinic and which regimens are being used. So we would first culture these platforms after receiving them from Columbia for 7 days, and then depending on the particular treatment, whether it's cisplatin, methotrexate, vincristine, dexamethasone, or a combination of drugs, we try to treat for several consecutive days and then have an off period and then treat again, and then evaluate growth of Ewing sarcoma cells 2 weeks after the termination of treatment. And so we've done all of this in comparison, as I mentioned, with two-dimensional culture. We're just cultured Ewing sarcoma cells. And what you see here is viability 2 weeks after termination of treatment. And we normalize everything to the growth rate of the Ewing sarcoma cells without any treatment. That's the controls across the top of this bar graph. And then you can see there is a concentration response. We've tested human Cmax and then kind of half an order of magnitude or an order of magnitude above or below. And you can see you can kill Ewing sarcoma cells with cisplatin. You can there's a clear concentration response with methotrexate in both models. Vincristine is very effective. Now, the interesting difference was with dexamethasone. Dexamethasone really is not supposed to have any effect on Ewing sarcoma cells, but it was toxic to the osteoblasts. And so Ewing sarcoma cells were dying as they were losing the support of the osteoblasts. And then the combination treatment was more effective in 3D. So there are important differences between 2D and 3D, but there's, the biggest difference to me is how much variability is there in the 3D platform, because it's very difficult to control the number of Ewing sarcoma cells that are being seeded into this model. So you have to have an effect that is really a pronounced effect, and it's very difficult to achieve statistical significance and have a very good titration of concentration responses compared to the 2D system. And you also may have some of these unexpected effects like we've seen with the dexamethasone. Another platform that's really a static culture but still has a microphysiological nature to it was a intestinal enteroid model that was developed jointly by scientists at Johns Hopkins University and Baylor College of Medicine. And this is a co-culture of different cells from the gut, and some of them are iPS derived and some of them are primary cells or cell lines, and these are propagated as enteroids. But then once you start testing them, this is really a transwell system where you have a monolayer of this co-culture, and then you can add drugs into the transwell and then measure permeability, or you can measure permeability in the reverse order as well. And, I'm not going to show you a lot of data, but just as an example, the published data is on the left from Johns Hopkins and Baylor College of Medicine. That we're looking at transepithelial electrical resistance, the measure of the barrier function of this particular model. And as you can see, the tier is going up with days of differentiation. That's what you need to see. And on the right is very similar data obtained by our colleagues in Houston. The transepithelial electrical resistance goes up as it should be going, and then the permeability to FITC-labeled dextran is actually decreasing. So again, the barrier does take a couple of days to establish, but it is a pretty nice barrier that you can test. And again, I will not belabor the point of the mountains of data that we've collected with this model system. We've tested 24 different drugs, and this is a graph of how we selected these drugs. There's actually a lot of data on CACO2 model versus human absorption in the gut, and we've tried to test compounds that actually have very good concordance with the compounds that actually don't have a very good concordance between CACO2 and human absorption. And all of these data will be submitted for publication in the next couple of months as we're still working through analytical chemistry on these data. Now, the other types of platforms that we've seen were based on perfusion, but it's a perfusion that is driven by gravity. So there are no pumps that are involved in this particular experiment. And all you have is, as the example on the left of the heart on the chip or cardiac model from University of California, Berkeley, you can see the model is pretty small. Two of these chips can fit on one histological slide. In the middle, on the top, you see there's about 50,000 cardiomyocytes that are tightly packed into a chamber, and then the perfusion is actually happening along the sides. So there's a passive diffusion of media and drug into the system. And then you can see on the top right that there are pipette tips that are filled with media, and then the media just flows from one side to the next, and then it equilibrates, and then you can replace the media next day or in a couple of days. And we unfortunately cannot show you the movies. These are the two slides on the bottom in this box, but you can see that these are different experiments, and the reproducibility of how many cells we can actually get into this chamber is actually pretty good. And the types of data that we're collecting is actually beating. So there's a video microscopy of these cells beating after a couple of days of culture in this chamber, and it's very well known that you can get cardiomyocytes to beat in culture in the traditional 2D system. And what you see in blue on the middle left is these cardiomyocytes beat in both directions. There's not a directionality of beating. But our colleagues from Berkeley have published on this model, and they've shown that if you pack them into this particular chamber, you force them to beat only in one direction, so that you can actually measure contractility, albeit we didn't do this in our experiments. And at the bottom, you can see that we can establish the same directionality of beating. If you see color, that means that the cells are pulling in one direction and they're not pulling in the orthogonal direction. And then you can test different drugs and concentration response. You can do kind of escalation of the dose in the same device, or you can do other time series or other types of experiments, and you can derive effective concentrations, and you can compare those to the Cmax or other types of experiments. And again, you can see that both us and the developers have published a couple of papers where they've compared their results to those of human Cmax for a couple of known drugs, and these were very similar, which gives confidence in this technology. That we have not published our data yet, but we get very similar data, which again, is a good result and something that we're very glad to see. Another model that is based on gravity-driven perfusion, albeit not with the pipette tips, but more of the reservoirs, and that particular platform is the vascularized tumor model from University of California Irvine labs. And this particular model, if you see on the right, their first generation was, a glass slide with actual chambers that were filled with media. They have since miniaturized this into more of a 96 well plate-like organization. There are actually 12 devices because each has a number of chambers that are actually constituting one device. But, this is higher throughput because you can actually work with 12 devices at the same time. So this is data from a publication from the developers. You can see that endothelial cells that are seated together with cancer cells form networks, and in red are endothelial cells, and they're very nice networks. And then in green are fluorescent cancer cells. And in this particular experiment, they had 4 different cancer cell lines that were seeded into their devices. And then you can add different drugs to this, passive perfusion system, and you can compare the cytotoxicity of those anti-cancer drugs, both in a 2D system and in a 3D system. So it's, not a technology that's very easy to establish. However, in our lab, we were able to successfully not only establish these endothelial cell networks. You can see on top is the phase contrast image, and then on the middle in red is endothelial cell network, and then at the bottom in green is the same device that has been perfused from one side with fitsidextran. So not only these are cords of endothelial cells, these are actually perfusable channels. And as you can see, fitsidextran entered on one side actually fills in the network of the vessels in this particular device. And again, there's a lot of data that we have generated that I don't have time to go into right now. Now, the last couple of devices that I wanted to show you are actually active perfusion or pump-driven systems. And again, there are additional complications with this. So the first example I wanted to give you is the Proximal tubule device that was developed at the University of Washington. And this device is based on a commercial chip from a company called Nordis that can be used a couple of different ways. And I'll show you on the next couple of slides an alternative way this chip was used. But in essence, this is a part of a nephron. This is not kidney on the chip. This is, doesn't have a glomerulus, but there are a lot of questions about drugs and chemicals and their effects in the proximal tubule. So it does, represent a very useful microphysiological model. And then once you actually, put this all together, it's a channel that is lined up with renal proximal tubule epithelial cells, and this is the channel that is perfused with media. This is the system that can be either enclosed in these particular enclosures that you can acquire from Nordis, and in the middle here you can see how this is all looking in the standard tissue culture incubator, you can fit about 24 of these chips, and one challenge with these models is they are not 24-hour or 1 week experiments. It takes about a week to actually get the channel established and perfusable, and then after that you can do this experiment for a couple of weeks and cells survive quite nicely. And so we both use the devices from Nordis and just the regular syringe pumps that are much more economical way of doing these experiments, and the results are essentially the same. So it's not how fancy your device is or your setup is how technically skilled you are, and also how well you can differentiate between different cell sources. So the results of this particular technology have been published a couple of months ago, and this is an open access publication. I invite you to go and find this and read it in greater detail. But one of the things that we were really interested in is whether there is a difference in where you get the RP text. So our collaborators from University of Washington have shared with us a couple of vials of more or less primary RP text that they have isolated from donor tissue at the University of Washington, and then we procured cells from Lonza, which is a commercial supplier, and there are other companies as well, and there was no particular reason we have used cells from Lonza, so I'm not vouching for that particular source in any way. And here, this is the data on gene expression. As you can see, these particular cells from both sources express kidney-specific biomarkers, both injury biomarkers and kidney metabolism and transport biomarkers. One important thing I wanted to point out is that the basal expression are dramatic for important things like KIM1. So you can see HAV-CR1, which is the KIM1 is expressed at a much higher level in Lonza cells, but then lipocalin, LCM2, is actually expressed at low levels, but still you can see that in more primary RP-tex versus ones that you can get from Lonza. But then what we did with these data, we actually compared gene expression profiles to human kidney cortex data from another NIH project called Tissue Gene Expression Consortium, GTEx. And as you can see, kidney cortex is on the top in red, and our two different RP-tex in culture, either 2D or 3D, are towards the bottom. So they're not really identical in their gene expression profile to kidney cortex. But what you can see is it's really doesn't matter how you culture these cells. It's the cell source that is actually driving the differences in gene expression. And then the figure on the bottom right shows that with different treatments that we have subjected these cells to, and then the gene expression profiling, again, you have grouping or clustering of cells or data by their source rather than by the actual treatment. And that's a very important point to recognize. Now, the actual experiments that we described in this study were, again, comparing and contrasting HIM31 and Lonza cells, and you can culture these cells for up to 28 days, both in 2D and in 3D. You can see the channel 3D in fluidic. You see these cells are elongated with the flow, and they're more haphazard and scattered in the 2D culture. And then, KIM1 levels, again, are much, much higher in Lonza cells than in the HIM31 RP-tex. And again, they kind of, steady up as you go on with culture. And this is, a very healthy culture if you're not treating it with anything. And we really aim to replicate what the developers have published. So on the right, you can see key data from Webb et al. 2016 publication in Kidney International. And what our collaborators did, they not only described the model, they also put this model to the test with respect to its physiology. So in the middle on the right, you see that they have looked at switch in vitamin D metabolism with, addition of calcitriol, and then also inducibility of cytochrome P450 24A1 when you actually switch the vitamin D metabolism. And at the bottom, they found that as they lower pH of their perfusion media, they actually see an increase in ammonia generation. So we tried to replicate these key elements of their publication. Plus, we also tested polymyxin B effects, and those are panels across the top and the middle ones. And you can see that polymyxin B was toxic to RP-tex in 2D in both Lonza and HIM31 cells, but HIM31 cells were very resilient to polymyxin B in 3D system when Lonza cells really died off in the 3D chip. And important point of biomarkers of injury, KIM-1, you can see it go up very early if you can collect enough perfusate in these models. But as you can see with Lonza cells at 48 hours, you see KIM-1 is actually going down, and it's not because there's less toxicity. There's actually fewer to no cells to generate KIM-1 and to shed that into the media. But more important or interesting findings were with vitamin D metabolism. We found that both Him31 cells and Lonza cells induced cytochrome P450 24A1 in 3D culture, not in 2D culture when we added calcitriol, but we did not see the change in metabolite formation with Lonza cells, but with Him31 cells, we saw a very similar effect as in the published publication. But with ammoniogenesis, we actually found an opposite result. So as we lowered the pH of the media, we saw a decrease in ammonia generation. So we overall concluded in that paper that the key elements as far as physiology, vitamin D metabolism, inducibility of the metabolic enzymes is reproducible. However, some of the experiments probably do depend very greatly on a particular donor of cells or a particular source of the cells that you get. But if you can, as you probably remember, we also had this, last tier, which is trying to define the context of use and trying to do more than the developers have done themselves as they were developing the technology. And what we've done, we've tested a variety of nephrotoxic drugs, cisplatin, gentamicin, and cadmium, and we have shown that there was a time-dependent and concentration-dependent toxicity that we can replicate in this model. And then, more importantly, we have collected a number of data points trying to understand whether this particular model can be used for pharmacokinetic modeling. And again, that particular data is still under review being submitted, but the complication of the PBPK model, PK modeling of this particular chip, that it's a single compartment. There's really not a blood compartment. There is not a filtration in this particular system. So we're only measuring reabsorption of sorts, kind of deposit or disappearance of a chemical from a perfusate. But we were able to replicate with cisplatin, gentamicin, and cadmium actually very closely the clearance and reabsorption when we combine both the data that we collect and a PBPK model that was developed for this particular instance. As you can see, cisplatin clearance to filtration rate is greater than 1 in vivo, and we have replicated that for gentamicin. It's about even clearance and glomerular filtration, and then cadmium is being, clearance is much lower than filtration, so it's being actively reabsorbed. We can establish that. And the conclusions that we drew from that particular study is that while tubular reabsorption can be estimated with this particular model, you really need a PBPK model to draw some conclusions about the overall clearance, but much greater confidence can be gained by potential end users if you actually have a tubular secretion component as well. And that's exactly the direction that our collaborators at the University of Washington have taken. They've developed the next generation of this model where there are 2 parallel tubes, and this is the model that we will be testing actually in February, March, and April of 2019. Now, another example of a perfusible system is 2 models of the liver. This one on the top was developed at the University of Pittsburgh by Lance Taylor and Larry Vernetti, and it's a Nordis chip that uses a different part of the chip. There is a chamber that is filled with cells, not only hepatocytes, but also other non-parenchymal cells. And you can see an image of how this actually looks in the incubator. The pump is in the incubator because the perfusion distance is pretty short. And we've tested both primary human hepatocytes from thermo and also iPS derived from Fujifilm hepatocytes. And it takes about 2 days to establish this platform. And then we've tested it for up to 14 days. You can sample media daily. You can also do imaging daily on this platform. The second one was from University of California, Berkeley, from Kevin Healy. And this is a device that is made in Dr. Healy's laboratory. It's actually a two-channel system. It's one channel divided by a membrane. So you seed cells, and in this particular case, only hepatocytes without non-parenchymal cells into one chamber, and then you perfuse through a different chamber. And again, it's an actively perfused system. It takes about 1 day to test, and we again tested the cells that developers tested from a different supplier, so primary hepatocytes from Lonza, and then the same supplier iPS-derived hepatocytes. And imaging is a big challenge with this particular model because the membrane is autofluorescent, and that really gives you a problem with seeing into the device. So just to show you a couple of data points, folks usually test the functionality of hepatocytes in culture by measuring albumin and urea nitrogen secretion. And you can see the dotted lines with thermal primary hepatocytes. These levels are low in 2D culture, and then in 3D culture, both ourselves and University of Pittsburgh collaborators who had the same batch of cells, we have very similar results, and albumin levels and urea nitrogen levels are very high and maintained over up to 2 weeks. And then with Berkeley device, we had a different source of hepatocytes, and we really had the opposite result. There was no albumin secretion, and blood urea nitrogen levels really went way down. So here, one cannot conclude whether it's a device or the hepatocytes from that particular donor, that particular source, that were the reason why this was not a very good result. But when you have the same type of cells, and in this particular case, Fujifilm Cellular Dynamics International iPS-derived hepatocytes with the same NPC fraction, we actually have found that we have a very similar result to the one we showed with thermal hepatocytes. And in this case, we could compare it across 2 different platforms. Again, the results are very, very similar. So it's not the device, it's actually the source of cells that is very important, something that we have found with the kidney device as well. But the functionality is not ending with just albumin and urea nitrogen, so metabolism is very important, and we've done a lot of experiments. One example I'm going to show you with adding tafenidine to the perfusion media and then measuring both tafenidine and formation of hexafenidine that requires metabolism by cytochrome P453A4. And as you can see in 2D on the left, in the first day of culture, we do have hexafenidine formation, but that metabolism really goes away as you keep culturing these cells. But in the 3D system, hexafenidine formation is consistent. As we add, as we change media and add more tafenidine, we actually form hexafenidine very consistently. And then with I-cell hepatocytes, very similar results. In 2D, there's metabolism over first couple of days, but then there's a very consistent metabolism over time in the 3D culture. And then with Berkeley, Lonza hepatocytes, not surprisingly, were not doing much with respect to metabolism past day 1. But then when you have I-cell hepatocytes and the Berkeley device in 3D on the bottom right, you see again a formation of hexafenidine that is very consistent, albeit perhaps at the slightly lower levels than in the University of Pittsburgh device. So our conclusions from this particular experiment, that 2D cultures, as everyone knows, are really not suitable for metabolism studies, but then these 3D microphysiological devices from both laboratories really maintain metabolic function as much as they maintain the basic functionality of hepatocytes. And it's, again, what we've seen before, the source of cells is very important, and CDI cells are something that is reproducible from the same donor. And as we all know, hepatocytes from that are primary will be exhausted eventually, and having a different batch or a different donor may reproduce experiment to experiment variability. So let me conclude with a couple of slides here. So what have we learned doing testing of microphysiological systems over 2 years? First thing that surprised me that we were actually able to do this. It sounds daunting to be able to transfer technology that is very complicated from different labs, but once you have dedicated staff and once you understand what the workflows are and once you have help from the developers, it is actually possible. And again, I'd like to appreciate both staff that we had here and also our collaborators who have gone out of their way to help us establish this technology transfer. It's also very important that all of our data are in the University of Pittsburgh microphysiological database. In over 2 years, we have run about 1,500 different chips and about 5,000, 2D experiments. We have collected tens of thousands of data points and hundreds of images. And all of this information will be available 1 year after we have concluded depositing the data into the University of Pittsburgh microphysiological systems database. So I invite all of you to access that site and start visiting that regularly, because the data will be released. Our kidney data, for example, is now in public domain. Now, detailed descriptions of all the phenotypic endpoints and equipment that's necessary and very detailed protocols. This is also something that we'll be adding to the University of Pittsburgh Microphysiological Systems database. This is very important for the potential use of this technology by end users, because it's not enough to just have a publication and then materials and methods section, or even a protocol from a developer lab from people who actually do this every day. So the detailed protocols that we develop would be potentially very, very useful. There are very typical challenges. Every platform is different, and we do not stop to be surprised how different these technologies are, and you have to have a very open mind working with any of them. But what we found is not all developers are open to sharing their technology, and that's okay. I mean, not everyone is willing to give their devices to someone else to try and test. Some of the devices and the quality, have to make them. There's a lot of material science, and there's a lot of lithography and printing that it goes into that, and the rigorous quality controlling of devices themselves is very, very important. Likewise, quality of cells that are used in microphysiological systems and devices is equally important. Sometimes these cells are not available, especially if the platforms were developed with primary cells. And then there are, different devices have different levels of technical complexity, and you have to be prepared to deal with that. But there are also a lot of opportunities. We are continuing this work for the next 2 years. We have 6 of our existing partners and 5 new partners who have agreed to continue working with us. And we have 19 microphysiological systems that we aim to test in the next 2 years, from 2018 to 2020. We do see that developers are generally interested in the feedback that we provide. And then a number of stakeholders, both folks in the pharma and the chemical industry, are generally interested in our experiences. And we are doing a lot of visits to talk to them about what we have found and how this technology can really be ported. But I really am, or feeling that I'm a representative of a potential customer. So I do have a number of thoughts as well to share with you. I do believe that this technology is extremely useful and very promising, and that bioengineering research and development needs further support. However, the applications of these tissue chips will be highly fit for purpose. And I do not believe that in the short term, the next 3 to 5 years, these tissue chips will really replace animals or even in vitro studies. I hope that some of these devices will be ready, but in reality, in the short term, we're not replacing animal testing. And for all of you listening, I think you have to, really, believe in this technology to start replacing data that you're used to from standard in vitro studies or from animal studies. But the developers also need to appreciate the need for portability and ease of use and reasonable cost of these devices. These are not easy to work with. These are not less expensive than traditional in vitro studies or even animal studies. And so where that balance is between the throughput and the cost of these complicated technology, I'm not sure we have a very good, comfortable understanding of that yet. And when people think that really it's the bridge between the developers and then the regulatory agencies, I think that first bridge is between the developers and then the users who will develop data, who will then go to the regulatory agencies. But the end users, companies, a lot of you are listening, really need to be ready to be thinking about these technologies, and they need to have the workforce that is ready to start working with this. Toxicologists or biologists may or may not be immediately comfortable using this technology. So thinking of who to hire and how to train staff is also very important. But then the regulatory agencies, the decision makers, both internally and externally to companies, also need to be educated about what the state of art is and what it can and cannot do right now. And the work of our center and our centers like ours is very important in this regard. But finally, what type of validation really we need to think about in terms of tissue chips? Validation is a very standard and very well-defined process for in vitro methods that replace animal tests. But tissue chips are far more complicated, and it's a technology that's still being very actively developed. And the real validation, the one that exists in Europe, where there is a network of testing laboratories and where there is a ring trial methodology, this is really not that is ready, in my opinion, right now for this very complicated technology, and again, technology that is still in early stages of development. Organic validation, this is kind of what our center has been doing. Sometimes felt like, very complicated thing, because these devices are engineering devices, and there are lots of connections, there are lots of tricks that you need to learn, and you really need to be ready for this. But finally, what I think really is going to take hold, and this is something where we're working with next, is the fit-for-purpose validation. The industry that are users need to define how they, what questions they think are ready for tissue chips, and then that information has to be embraced by developers to be able to develop technology that fits that particular question, rather than developing a technology that replaces a particular animal or an organ system. So I really appreciate your time listening to this. I'll be happy to answer any questions. And with that, I will conclude. Thank you.